I didn't realize that the deadline is so close. I thought there would be a seminar that just some time to think about it.
I mean, we've been advertising for it, so I guess the hope was that people would be reading
alert.
their emails and be thinking about it, but maybe that was a bold assumption on our part.
Right.
So
Just for some.
it's also possible that depending on how many applications, we could always extend the deadline if it's that people.
When is the deadline?
It's Friday.
Okay. So 2 days.
Yeah, but
I
the
was
hope,
gonna
we've
say-
been emailing, so the hope is that people have been reading the emails and seeing that we've been trying to promote this workshop. But
Yeah, just came back from LGBT conferences. These guys made quite a few impressive new announcements of the workshop.
Yeah.
Very fascinating.
So I'll spend some time at the end talking about all the new products that are coming out.
cool.
So you've seen the LGBT slides then, have you? Okay. Pretty much the
I put a
same, but
doorstop.
it will be good,
Okay, perfect.
good to introduce all the upcoming products to the folks that are going to be here.
Yeah, so this is what the video is going to look like.
Okay.
And I can send you the link afterwards if you want to see how it goes.
Sure, sure.
But I'll be basically screening your screen, and occasionally I can see you a little bit here.
Okay.
But most of the time it's going to be focused on the screen.
Sounds perfect.
Did Chris Andrew get back in touch with you about?
He said that he was going to talk to the dean and then
Oh, okay.
was there, do you know anything?
I think he's still interested in Mohammed Murtaza for the Shipley space. But I haven't heard anything else from him.
The
Well,
Shipley space.
I'm also interested from the point of view of new sequencing instruments.
Yeah.
Should be professor. I'm meeting with Andy this Friday.
Andy Henderson?
No, Andy Taylor.
Oh, Andy Taylor. Oh.
Basically, talking that I can do this.
Oh, okay.
See how it goes. Because Lumina is giving pretty aggressive trading.
Mm-hmm.
We'll see, it will be a shame if we don't use that.
Would we write a shared instrumentation?
It's
Or is this not?
useless, I don't think. We tried a few times, but in the Boston area, when everybody around is
Yes, sir.
way more,
Oh, welcome.
you need to prove that you have so many samples that like with the whole Boston
Okay.
capacity. we
Yeah.
can-
3 feet away.
Six.
6 now?
And hold your breath, please.
Yeah, exactly. My lab, I now have 2 people who formerly have it
What?
in the lab.
In your lab?
Yeah, I mean, I don't know that they haven't been tested, although Peter Ash's wife is the one that does the testing for Massachusetts. But they have, they both have symptoms.
And reason to believe that they actually have
I mean, it could be, I mean, you know,
true community spread.
it's not necessarily COVID-19 in terms of testing, you don't know.
Wow.
Dun, dun, dun. I know, I'm wondering, I just don't want I don't want people to miss out, but
Okay.
can you tell if there's people online maybe?
We've got 3 viewers.
Perfect.
Hello, viewers.
All right, I mean, I guess we can get started. People will trickle in.
Yeah.
Sure. Just in the interest of time. So I'll just give a brief introduction, and maybe Rosha, you can give a better introduction for yourself. But Science and Technology Advisor for 10x Genomics. You're just going to talk to us a little bit about ATAC-seq and maybe some interesting ways to analyze that data a little bit later on.
All right, thanks so much for the introduction. As Rachel mentioned, I am Rosha. I am a Science and Technology Advisor with 10x Genomics. So what that means is if you have experimental designs, you want to get started with single-cell sequencing, you have ideas, you can talk to me and then we will help plan you your experiments and give you the details of and answer questions that you might have. So today's talk will be mostly focused on looking at epigenomics at a single cell resolution. But having said that, I will go through there we go, our 10x genomics technology overview, so you know how we're enabling single cell gene expression, ATAC, etc. assays, as well as we have a spatial profiling product called Visio. So I'll briefly, just 2 lines about Visio once we go through the technology overview. The product deep dive for today is going to be single cell ATAC. Then there's going to be a very brief publications review, as in how other researchers are using our single cell ATAC technology to answer very interesting questions. We do have quite a bit of new products coming out, and we're actually very excited about everything that's slated to come out in the summer and rest of the year. So I will also spend some time going through some of the highlights of the new products that are coming out. So biology is immensely complex. We all know that, right? There's 6 billion DNA bases in the genome, each of our cell, but they're all differentiated. There's gene regulation. Certain cell types will only express certain types of genes. Going from DNA to mRNA, now we have more than 50,000 mRNA molecules that are expressed. And then going down to now protein, if we think about our immune system and all the antibodies that our immune system makes, it's in the billions. So it is very complex, and so far what we've done is, if you think about looking at this whole tissue, organ, a tumor specimen you have, if you take this brain, grind it up, get an average data point when you're doing bulk sequencing, now you're missing out the heterogeneity in terms of regulatory elements, as well as gene expression, protein expression. All of that is now turned into one average data point. This might actually not even be representative of what is actually happening in the tissue and the organ. So if we were able to look at this at a single cell level, then you're going to start teasing out all the heterogeneity that's within these cells that you have, different kinds of cells that you have, what different cells are doing in this system. So you're able to tease out all of this using single cell genomics. A day-to-day analogy that we like to use is, for example, this is your sample, you're making a smoothie, you have bananas, strawberries, you blend it up, you taste it, you get a hint of banana. I can taste the banana, but you're kind of losing everything else that might be in smaller portion in there. So what single cell component analysis lets you do is it tells you what is in that smoothie, as well as what proportion all of these different ingredients are in there. So this is a brief slide summarizing our 10x Genomics innovation engine. We started with our linked read assay in 2015. So essentially this was a way to use short sequencer, Illumina sequencing, to phase out long stretches of DNA information, fill out gaps in the genome, etc. And then in 2016 is when our single cell portfolio for gene expression, RNA-seq, launched. So all of these things you see 2016 to 2018 has been based on our single cell analysis. We've added capabilities to look at cell surface protein expression along with the gene expression from the same cell with our feature barcoding technology shown here, so on and so forth. And in 2019, end of last year, we also released our Visio spatial gene expression solution. So with single cell analysis, you take your tissue, you generate single cell suspension, single nuclei suspension, and then you can look at the t-SNE plots to see the different expression levels, different components, different subpopulations within your sample. But at that point, now you're losing the cellular context, the tissue context. So with Visio, what you can do is you'll take a tissue slide, put it on a tissue slice, put it on a slide, and now you can look at gene expression on the tissue itself. So it's also giving you additional capability to look at where exactly on the tissue these cells or these genes are being expressed. And in 2020, actually just a few weeks ago, we shipped our first Chromium Connect. So this is an automated way of running your single cell experiments. You essentially just put in your single cell, single nuclei suspension, all automated. What you get and at the end is going to be a library that's ready for sequencing. So it takes out all the manual effort you have to do in order to get your single cell data. And one of the things that's just shown here is a targeted gene expression, which is something that's coming out soon from 10x, and I'll spend quite a bit of time towards the end telling you about all the new products that are supposed to be launched. So we like building tools that researchers can use to answer exciting and interesting questions. By the end of 2019, we had about 700 plus publications that utilize the 10x technology. A lot of these were in high impact journals like Nature, Science, Cell, in various different kinds of research area. Cancer immunology, I've had customers who have worked with yeast, so this is applicable in a wide research area. So for our single cell workflow, regardless of what assay you use, so gene expression, immune profiling, single cell ATAC, which are all shown in these little colored boxes here, it starts with user supplied cells or nuclei or molecules. Then if you do library generation using the Chromium controller, what comes out of this is going to be a final Illumina sequencer compatible library, which you then sequence on an Illumina sequencer. And we also have analysis pipelines and visualization software that lets you look at your data. So what we have realized is data analysis can be a big bottleneck. You can do the experiment, then you can sit on it for months because you don't have the capability of, or have computational biologists who can look at the data for you. So what 10x does really well is whenever we launch a product, it's an end-to-end solution. We also launch software solutions that go with the product. The software solution essentially can be broken down into two. One is the analysis, the other is the visualization. So for analysis, this is Linux-based environment. It runs all the pipelines where you do alignment, quality control, etc. All of that is done with the analysis portion, so Cell Ranger DNA, Cell Ranger ATAC. At the end of this, you get web summary files that gives you a QC of how well your single cell experiment worked, etc. that you can, and you also get industry standard bed files, etc. that you get from sequencing data that you can then either dig down deep into or take it whichever way you prefer. Then we have the visualization software. So Loop Browser is our go-to browser where we can look at differential gene expression, differential accessibilities for ATAC solution, and it lets you at least get a visual of what your data looks like so you don't have that bottleneck. So today's talk is on epigenetics. I don't know how many of you are here are epigeneticists. So just to briefly go over what ATAC-seq is, assay for transposase accessible chromatin. So if you were to do an experiment like this where you're looking at enhancer promoter, CTCF, you would do a ChIP-seq against each of these proteins, so H3K4 trimethylation, K36, K27 acetylation, CTCF protein, and then you would do all these different experiments with a lot of input material, analyze it, and put it as different peaks on the genome, right? So with ATAC-seq, what's happening is we use a transposase, so TN5 like transposase. It is charged with two different Illumina adapters. So majority of our chromatin is heterochromatin. There's only 10% or so of our genome that's euchromatin. So once you incubate your nuclei with these transposase, wherever the DNA portion is accessible, it can go in and clip that DNA. So what it essentially does is it's like DNase 1 hypersensitivity assay almost. Wherever there's accessibility, it will go in, it will clip the DNA, it will drop these adapters. So essentially now you have adapters to amplify your genome and sequence it. So wherever you see peaks means, I'm sorry?
What's the frequency at which it goes in?
Frequency meaning how often does it clip or how well does it go into the nucleus?
How often does it clip?
So there is a titration that happens to figure out what the balance is. So because you don't want it to be over clipped, all of that chemistry is already taken care of in our system. So essentially if you're going from 500 nuclei to 10,000, you will use the same amount of transposase. So once you have this, we also have the capability of looking up transcription factor motifs from the Jasper database. So based on that, if certain peaks fall into certain transcription factor motifs, then you kind of know that there's a transcription factor bound there. Okay? So going back to how the technology works, the 10x technology, we have 3 different phases. So there's the solid phase reagent delivery. Essentially, we had a bar-coded primer library, and I'll show you what that actually means in the next slide. There is fluid partitioning, so we have partitioning oil. We introduce reagents that are water-based, so you can generate these nice droplets. And the idea is you'll capture a cell or a nuclei in these droplets that you generate. You collect the droplets you've generated, whatever assay you're working with, specific biochemistry happens within that droplet, so you're generating hundreds of thousands of nanoreactor chambers, and instead of doing the reaction in bulk, there's reactions happening individually, and then you do, after barcoding happens, you just pull everything together and proceed with it as if it were a bulk reaction. So this is what it looks like for our ATAC solution. We had these 10x barcoded gel beads. There's about 750,000 unique 10x barcoded gel beads, and what that means is inside of every of these gel beads, there's going to be oligos of this construct. You have the P5 adapter for Illumina sequencing that's required, or binding to the Illumina flow cell, and that's followed by a 16 base pair 10x barcode. So this is what confers cellular identity to the DNA fragments that come from your nuclei. And then we have this read 1N as part of what binds and what we need for sequencing. So every oligo that comes out of this barcoded gel bead will have the same 10x barcode. So the idea is you want to capture one nucleus with a gel bead, biochemistry, and then you have your DNA fragments that are barcoded. So that's what's shown here. You do your transposition in bulk, you have your transposed single nuclei suspension. This gets introduced in one of the channels. You introduce 10x barcoded gel beads. You generate these droplets where you capture a gel bead with a nucleus.
So, can I ask a
Yes.
question? So in this case, the transposon complex is diffusing through the nuclear membrane and getting in?
Yes, so you do the transposition actually in bulk. You will have your sample, either cells, you isolate nuclei, tissue, you isolate nuclei, and then you do the transposition in bulk. So all of this is happening in one tube.
Before the sorting into the beads?
Correct.
I
And then
see.
what you actually capture is a transposed nuclei with a gel bead.
Okay.
Yep.
So Russia, one of
Yes.
the announcements was that now you can do single cell RNA-seq and ATAC-seq
Yes.
from the same sample.
Yes.
How different is that?
That's my last bang at the very end. I will talk about the upcoming product. So yes, the new capability is both modalities from the same nucleus. Yes.
What you show is ideal distribution of beads and nuclei. Do you have situation where you have 2 beads per droplet?
Yes, great question. In 2 slides, I'll answer your question. Yes. So after the linear amplification happens here, at this point, the DNA fragments that you have are barcoded, so we don't have to keep them in all this oil emulsion droplet anymore. So we just clean it up, your nucleic acid is cleaned out, and then you just generate your final sequencing library here. This is just to show how the phenomenon works on the microfluidics. So essentially, the partitioning oil is here. You're generating droplets at this interface right here. These big round things are gel beads, and then you'll see a gel bead captured a cell.
If I remember right, that's incredibly fast. Like you've slowed that down many.
Most likely. Okay, so I'll answer your question about multiplets here. So this is what all of the single cell portfolio, the products that we have that run on chips, all the chips look like this. So you can run up to 8 samples at a time, so there are 8 sample wells. That doesn't mean you have to have 8 samples, you can run up to 8, so you can even run just 1 sample if you needed to. And each of these sample wells you can capture anywhere from 500 to 10,000 nuclei per channel. So this brings me to the multiplet rate question. So whenever, it's a closed system, whenever you put these chips into the instruments, you're approximately generating the same number of droplets. So if you increase the number of nuclei you put into the system, that just means there's going to be more droplets that will have 2, 3, 4, or multiplet nuclei captured. So for our system, there's up to 65% capture efficiency, so there's going to be some dead volume loss over time as you process your sample. So considering that 65% nuclei capture efficiency, if you were targeting to recover 1,000 nuclei worth of single cell data or single ATAC data, you would actually put in close to 1,600 cells into the system. And for this number, the multiplet rate is at 0.8%. And this does scale up linearly, again, just because I said it's a closed system, so the more nuclei you want to recover, the more you will put into the system, and this goes up linearly. So for example, for 10,000, this is going to be close to 8%. Okay. So this is just a very brief overview of biochemistry of what's happening. So the Transposition happens in bulk, so you're doing these in your 2 mil tubes, 1.7 mil tubes. Once you generate these droplets, this is where the barcoding happens. So if you have your transposed DNA, there's going to be a few different steps that at the end what you end up getting is DNA that's barcoded with the 10x barcode. So at this point, no need to keep them all separate. Then you'll add sample index. So you can index multiple or run sequence multiple samples on the same flow cell, and this is ready to go onto your sequencer. With single cell experiments come the question of how much should I sequence, how deep should I sequence. For the single cell ATAC solution, the recommendation is 25,000 read pairs per nuclei, and this just shows you here what the final DNA construct would look like in terms of sequencing. Okay, any questions so far? Yes?
How do you titrate the amount of transpososome so that you don't over-digest your chromatin?
So I'm assuming there is a curve, that was determined in-house that from this to this, it doesn't matter, this is how much you're gonna put in. So I know even if you go to the lower end of 500 to up to 10,000, there's really no issues of over-digestion. A lot of times what happens with transposes is it's almost like a heat sink. Once it goes in clips, unless it can recharge with another adapter, it can't really go somewhere else.
Right. Well, but I'm assuming those conditions were optimized on mammalian cells?
Yes.
So some of us use model organisms where the chromatin can be quite different.
Yes.
And if we want to do this on a mosquito or fruit fly, well, we have to re-optimize everything.
Not everything. So I've had users who have used planarians with our system.
Okay.
They did have to do a little bit of tweaking just because the genome is so much different and smaller than human or mouse, which is what our system or most of the work we've done is based on. And people have gotten some success. So if it's something that's not human or mouse or something that's similar genome size, you might have to do a little bit of tweaking.
Right, okay.
Yeah. Yes.
So 25,000, is it covering the whole open chromatin regions? Perhaps it's unnecessary.
So 25,000 is what we recommend. It usually is deep enough of a data to give you good signal. For gene expression, for example, we'll say, start at 20,000. If you're looking for a certain population but you don't see that separation or you don't see your lowly expressed transcripts, you might have to sequence a little bit deep. For ATAC, usually at 25,000 you get a pretty good resolution for what you need.
But it doesn't cover all open chromatin. Maybe technically that's a feasibility that produces a good signal, but you still underrepresent target regions.
So part of the data analysis when you call for the peaks is actually done in bulk. So you take all of the data, call your peaks in bulk, and then you determine what your threshold versus peak is. So I think a way to mitigate not have to sequence a whole lot is we do a peak calling in bulk. Okay, so I will briefly go into publications review. I have put the citations at the end of the bottom of each of these slides. So if you're interested in reading through these papers, feel free to do so. I do have quite a bit I want to share at the end with the upcoming products that we have. So I apologize if I kind of just breeze through these very, very quickly. So the first publication, this is actually one of the first publications that came out that utilized our single-cell ATAC solution. So this is from Howard Chang and William Greenleaf Lab. Will Greenleaf was one of the founders or people who pioneered the ATAC assay. So what they were looking at, so massively parallel single-cell chromatin landscape of human immune cell development and intratumoral T cell exhaustion. I'll show you some of the experiments they did with the T cell exhaustion at the end, but some of these figures that I'll present here are almost like proof of concept figures to show that what you get with single cell ATAC is pretty close to what you would get with bulk ATAC. So here we're looking at 820 lymphoblasts from mice. Up top there's actually another panel that shows you bulk OmniATAC track as well. So the 4 different tracks that you see here are for single-cell ATAC-seq, but they're just bulked together to give you bulk peaks. And then the bottom 2 panels here are randomly picked 100 nuclei out of the 500 and 350 nuclei that were present there. So essentially what this figure says here is what you see in your bulk ATAC data kind of is also recapitulated with your single cell data. So you can see these peaks that at a single nuclei resolution. So what they did next is something called computing z-scores. For ATAC peaks, it's pretty standard to do. So you look at where your peak falls, either transcription factor motif or distance from a gene promoter, and then you compute something called a z-score. So here we're looking at myeloid progenitor clusters. If you look at these, for example, GATA1, SPI1, KIT, all of these are myeloid differentiation genes, so it shows that there's higher z-scores here, so more accessibility at these myeloid progenitors, which is what we expect. One of the nice things that single cell ATAC can do for you is if we look at the IRF8 locus, which is important for immune cell differentiation, so at the plus 37 kb enhancer, you see this nice peak in the bulk aggregate data set. And then you see a lot of accessibility peaks throughout all of these different cell types that's been clustered. So if you look at plus 54 and plus 56, now this population here, you see the enhancer element is enriched in the plasmacytic dendritic cells, some here as well, but this is not a phenomenon these peaks are not found throughout all of the different clusters of cells that are there, as well as the same with plus 85 and plus 87. You have this particular group of cells that have accessibility at these enhancer elements, and not all of them have that. The rest of the clusters don't have that. So if you were to look at this data set in aggregate, you would actually not be capturing any of this heterogeneity and diversity, because all of that is bulked and averaged out in your bulk analysis.
Is it bone marrow or what kind of
Yes.
cell?
I will have to go back and refer. So they worked with PBMCs and blood bone marrow, or bone marrow cells, the two types of cells. So we wanted to also apply this to basal cell carcinoma. So basal cell carcinoma is one of the most common cancers in human. People with advanced basal cell carcinoma can benefit from PD-1 blockade therapy. However, half of the people who get on this therapy, it's not beneficial for them. So they wanted to test using single cell ATAC and try and figure out what could be different in terms of regulatory mechanisms that certain people with advanced basal cell carcinoma respond to PD-1 blockade, the other population doesn't. So this is site matched. So, and the sample type is pretty small, so this also shows that this product can be applied to very limited small sample size clinical specimen if you have that. So site matched tumor, you dissociate pre and post PD-1 blockade, you dissociate into single cell isolate nuclei, perform single cell ATAC, and then look at the tumor microenvironment as well as the tumor infiltrating lymphocytes. Yes?
Yeah.
Did you have a question? Okay, tumor infiltrating lymphocytes to figure out what might be going on here. So on this TC plot, what we see is when we look at the different clusters of cells that are present, this is from all 4 patients, you see there's a lot of immune cells that are listed here in green. You do see some stromal, so endothelial cells and fibroblasts, and then there are tumor cells. So what is very neat here is all the immune cells and stromal cells, they're kind of just all clustered together. But if you look at the tumor cells, they're actually separated and clustered based on the patient. So you see 4 different subclusters here for the tumor cells. So this shows that there's some sort of genetic heterogeneity within the patient that, all the other immune cells and stromal cells are, regardless of the patient type, kind of clustering together, but there is heterogeneity within the patients themselves. Going further down, so now they just looked at the tumor infiltrating cells. All of these different clusters, they're figured out based on transcription factor motif and the distance from where these regulators are for all these different kinds of different genes that are shown here. So they profiled about 28,000 TILs in terms of responsive patients and not responsive patients up top here. So in this population, what they found was there's actually follicular helper T cells, CD4 positive, and exhausted CD8 positive T cells. So the patients who responded had an expansion in this particular T cell population.
Question: are all these figures coming out of the Loop browser, or are these generated by the lab?
So these have further downstream workflows. So I think Chrome var is one of the standard single cell ATAC solution, or a single cell, not single cell ATAC, in ATAC in general to call peaks, etc. So they've used some standard tools that are already available, but they've also done some further work.
I see.
So, yes.
The tumor cells of the basal cell carcinoma, you show a bulk of the cancer cells. Are those the ones that were susceptible to PD-1 or the ones that were resistant to PD-1?
The ones here?
No, in the previous one. Yeah, those, the 17 to 20.
I actually don't recall. I'll have to go back and check on the publication.
When you say that they're responsive to treatment, basal cell carcinoma is pretty much just you cut it out and go. It's not an aggressive tumor, so there's no like metastasis, or so when they say the response rate, they're taking the tumor out and then.
I'm not actually sure what they look for.
Just
I
curious.
guess the
questions.
real question is if you have susceptibility to PD-1 and resistance to PD-1, maybe you have two different populations of the cancer cells. But this technology doesn't show you that, only tumor cells, and that's it.
So
right?
you could further dig down deep into just these cells and try and figure out what might be differences within that subpopulation. Yeah, so this is compared to everything that was part of the tumor that was dissected, but you can definitely just hone down into just the tumor cells.
So, let's say you had, you're putting in cell types, like, so you're doing the brain, let's say, your brain, and you have a whole bunch of, in your cortex, a whole bunch of neuronal types, and I put in some, virus, and I get a response only in some cells and not in others, even though it's everywhere. And so I do the ATAC-seq, and I see a signal in those. Can I then go dig down and, like, since the ATAC-seq is done on top of the single cell RNA-seq, then kind of determine what some of the characteristics of those cells? To say like, oh, it's this type of neuron or that type of astrocyte or something?
Yeah, you would have the capability to do that. So this is just doing ATAC, right? There's no RNA-seq involved yet. The next paper I'll show you, they actually split the samples into two, do RNA-seq on one population, ATAC on the other, and overlay the data computationally. But with the product that's coming out where you can look at the nucleus and ATAC from the same nucleus as well as gene expression, I think that would be what you would be looking for, because then you're getting dual modalities from the same cell. Let's go here. And then there's some more involved bioinformatics that you can do where you can look for different cell trajectories, how they move in this UMAP space, how they might be differentiated, etc. And then you have the T cell lineage. So essentially what they're looking for here is when you go from naive T cells to the exhausted population or the follicular helper, what kind of transcription factor motif excitability changes. And there are certain factors that are shared between both of these types. So based on that, there's just this general anti-tumor T cell response model that they come up with, that these factors are involved in different parts of the T cell differentiation when you're looking at these TILs. So usually when we're talking about single cell ATAC data with gene expression, if you see accessibility, which is shown up top in the panels here, versus gene expression, there's almost a concordance. So if you have accessibility, you have expression. But there can also be cases where if you look at the ELF1 locus, so you look at transcription factor accessibility, so it's up in T cells. If you look at expression, it's very broad within all the cells that were in the system. And then if you look at TBX21, where the accessibility is very broad within all the cell types, but if you look at gene expression, now it's showing up in the CD8 and natural killer cells. So this is just to show that, you know, you can take a screenshot with ATAC, you can do gene expression, but just because you have accessibility doesn't mean it might be transcriptionally active. So to overcome one of those issues and do a multimodal experiment, so this is again Greenleaf's lab. What they did was they took this healthy bone marrow and blood, and they split the sample into two. So the top part is site-seq. So site-seq essentially means you can label cell surface protein, and you're able to capture which cell surface proteins are expressed along with the whole genome transcriptome. So you get two different information but from the same cell using this part here shown in blue. And then the other half of the sample that was split off, they performed single-cell ATAC-seq with this. So this was applied to mixed phenotype acute leukemia, or MPAL, we'll just call it MPAL. They have cell populations that express myeloid and lymphoid lineage, so they're very heterogeneous. What is being shown here in these two different panels is essentially if you do single-cell RNA-seq with site-seq and single-cell ATAC-seq, you're able to capture all the different subpopulations you would have using both of these assays, so they're almost complementary. So here is where some of the computational magic comes in, and I call it magic because again, this was samples that were split. So now they're trying to computationally layer these onto one another to be able to say that, you know, this is what gene expression, protein expression, and ATAC looks like. So what they do is they actually use healthy bone marrow and blood and generate almost like a baseline. And then the idea is using the transcriptome information and the accessibility information of these healthy cells, you try to figure out what might be going wrong in these mixed phenotype acute lymphoma, leukemia, sorry. So they call it the Leaky semantic projection. So healthy bone marrow is here, this is what it should look like. Here's the computational stuff where they apply the LSI projection. And this is an example of where they look at different patients. They're projected by mapping the cancer cells to the nearest healthy relative cells. So all of the different cell types are shown here. Based on that and your ATAC data that you get, you can also look for which transcription factor motifs are differentially expressed or accessible within each of those clusters. So here's a list of transcription factors. One of the one that comes up is RUNX1. And previously, this transcription factor has been associated in different kinds of leukemia. So they decided to go with RUNX1, and they, just by searching the peaks, and they found that there's about 700 putative regions where RUNX1 can bind. So what they ended up doing is they went to the TCGA, looked up for the gene expression profile for these MPALs, and these two plots, or these two lines here, show a survival curve. So the one in the red is if there is a high RUNX1 target, your survival rate is much lower than if it were lower RUNX1 target. So this is essentially to show that you can use single-cell and multimodal experiments to kind of get to these very molecular biology-driven phenomenon that might be occurring. Okay, so again, if you guys want to read these papers, they're pretty good. This one's a little more computationally involved than the first one, but all of the paper citations are there. From that, now I'd like to spend some time talking about our new products that are going to come out. So in terms of the single-cell portfolio, what we want to do is we want to be able to help you increase your scale, your resolution, as well as make sure the products that 10x is putting out is accessible to users who want to use it for different scenarios.
As
yes.
you go along, this might be painful for you, but can you tell us kind of how much each product costs, so I can get a sense of you know, how it relates to budget? I know these are expensive, but
I might have to.
Just the ballpark.
That's a tricky question. I might have to have Dan email Yuri a list.
Say, maybe we have like a developed spreadsheet of some sort just to give you a cost breakdown.
I have some idea about what.
Yeah, if you guys can give us some idea, so when I look at it, I have a sense on, you know,
how much you're gonna go into debt.
Yeah, I mean, what it costs for, I mean, I realize you can multiplex and things like that, so.
Yeah.
Yep, I can
And
have
I
Dan.
realize you don't want to, or you're not allowed to, you know, or whatever. but-
It's not directly my territory to talk about numbers, so that's why I'm a little bit hesitant, but we can get you recent
But,
numbers and you
it
can
makes
get
it- It's
them.
important to know ballpark, to know, have a sense on how it goes into our, you know, existing budgets or future.
Okay. So first, I'll focus on the gene expression and immune profiling solution. So one of the products we're slated to release sometime this summer is going to be targeted gene expression. So this will allow for more flexibility, higher scale of what you're doing, and also more applications and more experiments if you're doing targeted gene expression. So this is going to be compatible with our single cell gene expression, so that's 3' RNA-seq, single cell immune profiling, 5' RNA-seq / immune profiling for TCR/BCR, as well as the spatial gene expression or Visio product that we have released. And all of this, if you already have a library, you did feature barcoding, all of that is going to be compatible with the targeted gene expression.
So if I have an existing library, already set up, I can then apply this summer.
Yes, so the way you're doing the targeting is you will have a final 3', 5', whatever library you have,
Yeah.
and you enrich off of that final library. So if you wanted to go
Yeah.
back and do unbiased whole transcriptome analysis, you have the option to do that, or if you have a panel and you're like, I'm just interested in these genes, you can do that as well.
So
Yes.
what is the advantage of the targeted application other than coverage, or is just coverage?
Mostly the cost. So if you look at the sequencing cost here, so for the example shown here is for 8,000 cells at our current recommendation for whole unbiased transcriptome, 20,000 reads per cell, you will sequence about 160 million reads. So if you were to do targeted genes, our panels have about 1,100 genes in them. This cost is reduced now by 80%.
And so what does that correspond to in money?
Well, 5 times cheaper.
5 times cheaper.
What's the
For
starting?
sequencing only.
And for the pilots grants that we're doing through the GSI, those are 4 samples for about $10,000 of an experiment. But this is only for specific targeted gene sets that 10x Genomics has already defined, or what if I want to go after like specific viral transcripts that are currently they're missing in some of our experiments that we want to enrich just for those viral transcripts within the cells that
Right.
we're studying.
Yeah, so initially we are releasing 4 different panels, cancer, immunology, neurology, and then a pathway panel that has different markers that get expressed certain ways, about 1,100 genes. But you will have the ability to do custom panels, and we have a design tool where you can go upload your files and do custom panels as
Okay.
well.
Okay, cool.
So again, if you want to do whole transcriptome, you will get to that point. You can save your library. If you want to do an unbiased approach, this is an exploratory research, you don't really know what you're expecting, you would go with whole transcriptome. The sequencing cost is a little higher, but if you know what targets you're interested in, then your sequencing cost is going to be much, much reduced.
It's just sequencing a fraction of the whole experiment.
Yeah, because for now, what happens is you're still generating the whole transcriptome, unbiased transcriptome, and then based on, you can generate the library, the 10x library, and then you can decide if you want to do targeted or if you want to do whole genome. So these are just some examples to show that the targeted assay works as well as what we see with the whole transcriptome analysis. So this at 100,000 reads per cell. PBMCs, again, you see all these different clusters. Then what we did was we did an in silico analysis of where we just pulled out a subset of the reads to just the targeted genes. So you can see this is still somewhat similar, mostly similar to what you would see with whole transcriptome. And then at 10,000 reads per cell, this is still preserved. So you see all of these clusters pretty much look the same. You can even go down to about 2,000 reads per cell, and you would still be able to discern most of these clusters that you see.
So
Okay.
you could do the targeted read with like combining RNA-seq and ATAC-seq?
It's way advanced, but I think, yeah, technically, but I don't know, we don't actually know all of the nitty-gritty details of what the different steps are, but we can wait and watch, but something to look forward to for sure.
Theoretically, if you generate a whole transcriptome library and the tech library, and then you can apply the targeting approach to it.
I will say the ATAC is based off of nuclei, so if you're
Right.
looking for a whole cell, you're not going to get the whole cell transcriptome, it's going to be nuclear. But I'll get to that. Okay, so that's the first one. Targeted panels, again, there's going to be 4 preset panels, but you also have the option to design your own panels or spike in your own genes if you wanted to. The second product that's coming out is cell multiplexing. So with, you guys have probably heard of cell hashing. You can use different barcoded cell surface antibodies, proteins, antibodies against cell surface proteins. So the idea is to maximize the number of cells you can put in a 10x channel and get data out of it. So now we have our own way of doing that. Very flexible design, gives you high throughput. Important is that it's species agnostic as well as compatible with nuclei, and we will release a software that's going to be fully supported with cell multiplexing. So initially it's going to be for the 3' single cell gene expression and then the 5' immune profiling solution. It's based off of a lipid. You can do nuclei and cells. So for now, you can go up to 10,000 cells per channel. With this, you can go up to 20,000 cells per channel for recovery. And within one of your samples, you can multiplex up to 12 different samples. So if you had 1,000 cells from 12 different samples, you're able to multiplex all of that into one single 10x reaction. Okay?
So what's the timeline on that?
Summer. Yeah. Summer of this year. So this is just to show, we took PBMCs, we separated them into 12 different tags. So this is just gene expression based on expression profile. You see all the different clusters you expect to see with in PBMCs. Now we're showing you clustering based on gene expression profile, but these are all tagged with the different colors for the 12 different multiplex lipids that we used. And then when you do the individual tag assignment, you see that there's really no bias for a certain cell type. All of these look pretty good. There's really no bias for one tag versus the other. Individual tag assignment shows that they're pretty good. Okay, so we released our feature barcoding capability, which lets you look at cell surface protein. And then customers said, but I want to look at intercellular protein. What are we going to do about that? So now we're working on being able to also look at intracellular protein and not just cell surface protein. So I'll show you a very brief example of what we've been doing with our partners. So this is a proof of principle experiment that was done by our partner Cell Signaling Technology. They took GERCAT cells, just treated them with modulators, kinase inhibitor, 2 different kinds, activator, and untreated cells. So when you take this sample, you do gene expression profile, you don't actually see a lot of distinction between the different kinase activators or inhibitors or even the untreated. It's kind of all here. So it shows at the gene expression level, these post-translational phosphorylation, you can't really capture that information. But if you look at the same data, but now based off of intracellular protein expression, you see there is separation between the 4 different samples that we have here, and that you also see that for the 2 different activators, the salmon and the blue, you kind of also see a separation between those 2. So this shows that instead of just looking at gene expression, if you're able to look at intracellular protein expression, you can get this kind of information as well. So this is pretty promising.
So are they using antibodies, or what's the? I feel like I'm missing something about understanding this completely.
Antibodies, yeah. So for now, cell surface protein just has to be in the outside.
Yeah.
Now there's a fixing step.
Yeah.
So and then the
so
antibodies.
They're using some sort of antibody that recognizes a phosphorylated sequence of some sort.
Right.
So broad.
Right.
What's the expected number of proteins that you be able to read in this method?
So we say hypothetically it's based on the 10x barcode. hypothetically you can have billions, but the limitation comes to how well protocols work, because this will require cell fixation, and then that means, you know, RNA can degrade, etc. So all of those things we're working out on. Hypothetically, you can do as many as you want, but then it's also going to come down to how well the protein or the antibody works as well, right?
But it will be based on a panel of antibodies for those proteins known a priori. It's not like an unbiased swing on the protein.
Yeah, so those are, I think, details we're still figuring out. You know, BioLegend is one of our partners. They started with single antibodies. Now they have panel of antibodies that can go into the 10x workflow. So I think all of those things will get figured out as we get closer to launch.
But the
Yes.
antibodies have to bind things that are on the membrane?
So that's what is currently supported. So these are going to be antibodies that are going to be intracellular, and we're still working on finishing off the product so it's marketable and it can be in your hands soon.
So after fixation of the cells, these antibodies somehow go into the cell without.
Great question,
compromising the
because
cell?
I don't know the details of the protocol yet either. I've only seen bits and pieces
It's
of the
like
data.
Shakespeare in love.
Yeah. One of our other partners is BioLegend. So BioLegend is they make the antibodies with the oligo tags, so you can capture with your 10x experiment. So these are cell surface proteins, different antibodies shown here. Antibody signal, RNA signal here. For cell surface protein, you know, it works pretty well. We see good concordance, but there are also some examples of intracellular protein where we see similar concordance between the antibody and the RNA. So these are all things we're working on and should be released in the next few months. Okay, so this is what I think you guys are interested in, because we talked about this earlier.
And the cost is $1.01 per experiment, or what?
For the antibodies?
No, sorry.
Okay, so the next one is ATAC gene expression from the same sample. So the second publication I reviewed, you know, you take your sample, you split it into two, you do single cell gene expression, you do single cell ATAC, and you can infer linkage. So for example, you know, the yellow cluster here, probably computationally you can overlay data, infer that there are similar sample types, but still at this point it's not coming from the exact same cell or nuclei, right? So what we want to do with a single cell ATAC plus gene expression solution is you will take your sample, you'll get your gene expression and open chromatin information from the same nuclei. So this is going to be a true linkage. You're getting dual modalities with that. This is just a slide to show that up top is the gene expression. This is to our single cell gene expression V3 versus comparing it to the new product that is slated to come out. And if you look at the UMIs and the genes, median UMIs per nucleus or median genes per nucleus, you see that they overlap pretty well. So this new assay doesn't really infer or doesn't affect the sensitivity of the gene expression assay that we already have out. For ATAC, yes?
If you're doing an experiment, you know, to what extent do you have to use replicates?
That's a great question. So single cell field is expanding pretty quickly, and I think that's a debate a lot of people have. You know, initially you could do a few single cell experiments, it would be published, but now like reviewers are getting more and more rigorous. So, and then the cost comes in play too, right? Because how many biological replicates do I have to do? In terms of technical replicates, you know, in terms of the 10x reagents, everything is very good. Like the correlation is 0.99. So there's not much variability from one well to the next.
It's the biological
Right,
replicates.
right. So those are some things to think about as well. So the bottom panel here is looking at ATAC fragments, and compared to our current ATAC product, this is also pretty comparable. So this is going to be very exciting. Like I want to see how people end up using these, and you don't have to do all the computational stuff to overlay data on one top of another.
This is all in that Loop browser.
Yes, so every time we release products, so the Loop, because now, for now, it can do cell surface protein and gene expression overlay those. So because this would mean you would have to overlay peak information with gene expression, so there's going to be an updated release.
And is that Loop browser do we need to buy a license, or is that
No, all of our software is open source.
Okay.
There's no charge to customers. You can just download and use them.
Oh, okay.
Yep. So the Visium platform, some of the cool things we're doing with Visium, I didn't talk about this a whole lot, but I'll still give you an idea about what is coming. So Visium was only launched about November of last year, so it's been around for 3 months, but we're already looking at adding more feasibility to Visium. So the targeted gene expression, targeted panels that I talked about, that's going to be compatible with Visium. We're also going to add immunofluorescence, feature barcode technology, and an FFPE solution. So right now, Visium is compatible with fresh tissue that you embed in OCT and then you do tissue slices. But the biggest question is, my samples are FFPE. We want it to be FFPE compatible. So I'll show you a quick slide about how we're going about it and what the data looks like. So let's just go through these. So this is what feature barcode antibodies are like. It's an antibody that has an oligo that you can capture with the 10x barcode. It's going to be the same for gene expression and the protein, so that's how you know it's coming from the same cell. So if you look at immunofluorescence on Visium for CD29, CD4, these patterns look pretty close to what you see for feature barcode for CD29 and CD4. And this is just the gene expression shown here. So there's going to be a lot more information coming out about this as well. In terms of FFPE, so here is standard Visium. We're looking at fresh frozen tissue. This is all the genes that are shown in the different spots, versus here we're just showing an example of PRKCD where it's expressed. If you were to use the standard Visium on FFPE, the signal's not that great. So you can see there's just almost looks like random spots. You don't actually see anything here for single cell gene expression. We are modifying the protocol, and we will be releasing this at some point with the modified protocol. Even with the FFPE sample, you get some really nice signal, and you can get the clustering out. So this was for 3-month FFPE tissue. We're also trying out different time points to see how the sensitivity might drop, etc. And with the modified protocol now, you can again get signal from this part of
the gene coverage in each pixel?
Gene coverage as in how many? So,
mRNAs?
I'm sorry?
How many mRNA?
So the biochemistry for the Visium is exactly the same as our single cell 3'. What is going to be different is there are these spots that are on the slide, and they have all millions of oligos on each spot. So the biochemistry wise, it should be the same as our single cell. Yes?
Just to be clear, every spot is not a cell, right? Or is it?
So it's going to depend on your tissue type. So from one spot to another, a spot is about 100 microns. The distance between the two spots is about 50. So if you have a large cell, it can span 3 different spots. So it's not single cell. It can be anywhere from 1 to 10 cells, but that depends on your tissue. One thing that Loop Browser can do is when you see this data here, you have your gene expression data and you overlay your H&E stain. You can kind of decrease the opacity so you can figure out how many cells or nuclei are in those spots, so you can get that information from Loop. Okay. Yes.
Do you have to image? Is there an imaging step?
Yeah.
Did I miss that?
So no, we didn't go through a lot of the Visium today for it
okay.
because we're talking about ATAC, but you do an H&E stain and you
First?
take that image, and then you go through the biochemistry, and then computationally you overlay your H&E image with the gene expression information.
Okay.
Yes.
Would it be correct in assuming, like, in brain, you could go and use a workflow to, like, look at, in the Visium, look at the individual spot and say, oh, I have transcripts that are both glial and neuronal, therefore I know I have more than one cell type in this Visium spot? Is that right?
I think you would be able to do that. So I think with brain, mouse brain can easily fit.
So you know kind of what you're looking at, like how things compare among spots. Is it just, you could just be looking at neurons, you could be looking at neurons and glia, neurons and oligodendrocytes or microglia.
Yeah, so I think that depends on how you section your tissue. It has to be, slim enough to fit almost on a 1 cell layer, because that's what you want to capture. Does that make sense?
Yeah.
Because if you have too thick of a tissue, you're actually just capturing what's on the bottom that's touching the slide, and you're not going to see what's up top.
Mm-hmm.
Okay.
But
Right.
also width, 100 microns could easily cover more than one cell type.
Yes, so 100 microns is the diameter of the circle itself, but actually the capture area is 6.5 millimeters by 6.5 millimeters, so it's actually pretty small. So a mouse brain can fit in it, but if you're working with bigger samples, you have to section different part and put them on different capture areas. All right, so Visium, FFPE coming soon. And then at the very end, I want to end with 10x Genomics Cloud. So that's something we're going to introduce sometime midway this year as well. So what this will enable you to do is, it just means you can do cell ranger analysis, base ranger for Visium, loop analysis, all of that is done in the cloud. The computation time is going to go quite a bit down. There's a lot of different aspects that play into that, including improving our pipelines, etc. Standard analysis is going to be included with reagent purchase. I don't actually know a lot more details about what's going to happen if you're doing a 5 million cell experiment, how those things go, but at least the standard analysis is going to be included. So we're trying to reduce. Even now we say we have Cell Ranger, we have Loop Browser, it's free to use. Sometimes there's still that hesitation, or you don't have the infrastructure. How do I set this up? How do I get going? So we're trying to reduce that, take that out of the equation, so you can at least have your data initially analyzed. Okay?
What do you mean by zero stress?
Zero stress, I'm not really sure. So as things progress through the pipeline and newer products are coming out, there might be release dates, etc. If you want to keep in the loop, I would just recommend going here, and you can even sign up for an email for a particular product. So if there's updates, you get those emails as well. For your local team, so Dan Harkness is your sales executive. I think you guys will mostly be working with Yuri, so Yuri already knows Dan to get quotes, etc. Nicole is your FAS. She's not here today, but if you guys have any questions, want to talk about experiments, etc., you can email any of these people here, and would be very happy to help you out. All right. Any other questions? Yes.
Yeah. I don't know if I understood this correctly. Do you have plans of releasing a platform where you can do ATAC-seq and RNA-seq simultaneously on the same cells?
Yes.
Yes?
Yes.
That's
Yes.
what we're saying.
The last one?
Not the last
one. Before
one.
Visio, right?
Yeah, so the one where I talked about, getting true linkage instead of inferred linkage. This one right here. Yep. So
Those are the same cells, like the information transfer. Yeah.
So it's going to be nuclei based. You will isolate nuclei, but both of these information is coming from the same nuclei. Yes.
All right. Do you have a timeline?
Summer of this year. Yeah. So we say summer. Sometimes it can be quicker. Sometimes it can be later. Like, things are changing every day, so it's hard to give a date. But sometime this summer.
Awesome.
Yeah. Cool.
Great, thank you very much.
I think Rachel has a few
Yeah.
slides to share, and if anybody wants to stay back, I can quickly show you how Loop Browser works for ATAC data too, if you're interested.
Yeah.
Are you on the back too? So
Yeah.
let me just give.
I just have a few slides about the workshop, so I don't know if people were staying because they were interested in some of the details of the workshop. If not, then don't feel like you need to stay for this part.
The software is not on the cloud yet, is it?
Not yet.
Okay.
Yeah, so right now, Yuri already has it installed somewhere, so if you're using the same resource, you probably don't even have to download a local copy. If it's like within the View cluster where you do data analysis, but you can easily download it. You can't really run it, technically you can run it on a computer, but like the computing power needed is crazy. So
yeah.
people usually do like Amazon Web Services and get a Linux
Or whatever we have on
environment.
the View
Yep.
server. The cluster
And
is
yeah,
used.
Cell Ranger's
Yeah.
probably
I don't
already
have the right
on there.
adapter
Right.
actually.
Do we see? Oh, I was using that one.
No, I know, but this I don't have the.
Oh, I see.
I don't have that.
This? Will that work?
No. I need the Thunderbolt.
I don't have it. I'll try. How about this?
Yes.
That work?
I think so. There we go. Look at you. Is that yours?
Yeah. Thanks.
So for the 1, 2, 3, 4, 5 of you that were interested in the workshop piece.
And 10.
Oh,
live
10.
stream
Oh, okay,
audience.
perfect. I'm not sure if it's clear at this point. I found it a little bit confusing initially. That the goal of the workshop is really just to support additional research for academic people who are looking to expand their research capabilities, I suppose. So Nelson actually started this, 2 years ago? Before
Last year. No, last year.
Last year, before I became associated with the GSI. And so the main goal was to move from pilot seed grants to this kind of supporting technology and actual experiments, things that could further support your research. And so we've switched from that format, so skipping the seed grants and going straight to supporting these research initiatives. And so all this information is actually on the GSI website. So anything that I have here, I think we've posted it on the GSI website as well, so you can go and take a look at anything if anything I've mentioned is unclear. But I just wanted to give a quick, I didn't realize those were automated. A quick timeline for what were the expectations. So I mean, at this point, the goal was to have the workshop competition submissions be completed by Friday. So the idea is to generate a proposal that takes advantage of some of the resources from 10x Genomics to do these single-cell RNA-seq experiments. And so the application is very short. It's 2 pages. You can submit it to the GSI website, the GSI email address directly, and again by this Friday, March 13th. It's possible that we will accept them slightly later than that if they're Monday at the very latest, but yeah. But Friday would be excellent, just because we have to find, we have to enlist, we have to see how many applications we get, how many reviewers we're going to need to make sure that we can get everything done in a timely fashion. And so the goal is that the awards for those proposals will be announced in April of 2020. And the hope is that you'll have samples can be submitted as soon as April, but no later than June 1st. And I believe that's what we did last year, and we're still on for this year as well. So.
Because the fiscal year turns out.
Exactly, we were sort of on a budget timeline, and so that's why the dates are dictated the way they are. So the proposal.
Is that for the scRNA-seq or scRNA-seq plus a TAC-seq, or?
Whatever you're going to do needs to be ready to go by then.
Yeah.
So the goal of having Rosha tell us about the attack-seq was just so people have an idea of what they could do. So it's not just limited to certain aspects of single-cell RNA-seq. If some attack-seq is something you're interested in, and it's something that we're capable of doing, it's something that we can include.
Something to keep in mind that if you're doing attack-seq and RNA-seq, it sort of doubles.
It doubles, exactly. So
it doubles the cost.
Yes.
Doubles the cost.
Which means that GSI is unlikely to cover all of those things in one proposal. And so, so that's the goal for the submission of the proposal, and also when you would have to have samples ready by. And so there are some contingencies on this. One is that the goal is that you're going to present whatever you find if you're awarded one of these grants or research supplies. Is that you're going to present at the GSI symposium in the following November. And so just considerations for your proposal in general. The goal is that it can be exploratory, right? We're not expecting that you're going to have a really strong hypothesis-driven experiment that is going to cover all of your needs in one proposal. The goal is to make it something that is compelling scientifically, just in the sense that you're asking a good question, but it can be exploratory. Obviously, there's only 4 samples that are covered by the proposal, so we don't have this grandiose vision of this really complicated experimental outline. So keep it simple, but exploratory is fine. It doesn't have to be hypothesis-driven. Again, as I mentioned, the goal is really to generate preliminary data for some sort of future experimentation, and to help Ben figure out how much money he's going to need to budget in for this experiment of all the things he plans on doing. So again, as I mentioned, it's limited to 4 samples, so the ideally 2 experiments, 2 controls, but I guess in the end, if it's your proposal, you can pitch it however you want. 4 samples. So the pilot's the goal is to capture about 2,000 cells per sample. This includes the library prep, and the goal again is to target about 50,000 reads per cell. And I assume, Yuri, that that's still...
Yeah, they say 20,000.
I saw that,
but
but
they also say that going from 20 to 50 still might increase the amount of
exactly.
GSI.
So the range would be
It's all
20 to
sample
50.
type
Yeah,
dependent,
exactly. And so capturing more cells, we had previously mentioned that this would be there would be an additional cost incurred to researchers who had specific goals of capturing way more cells for whatever reason. So those are sort of the goals we have outlined, but I think if anything varies from these kind of general specifications, that you should either chat with us at the GSI or Yuri about what those expectations might be. And obviously feel free to jump in if you guys have anything to add. I think the main goal, I think the thing that we experienced last year too with the single cell sequencing was that people had to make sure that their cells were sortable in the time frame that we need to submit the sample. So that includes making sure that the cells, you have single cell suspension and that the cells are actually viable. And so if there's a need for additional protocols for dissociation, I think at this point there's a number of labs who have done this successfully through BU. So if you're struggling with the protocol itself, feel free to reach out to any of us and we can help you with those protocols to make sure that you have not only the right amount of cells, but also the viability that you need. And so
since last year we also had decent success with sequencing single nuclei,
yeah,
which might also
be useful
increase
to
the possibilities of
in case your cell's hard to sort.
exactly.
you
can do
or
nuclei as well.
Maybe even frozen tissues or something.
Yeah, I mean that was what we did. We did it from frozen tissue. We isolated single cells, and we had good viability for that. So for anybody who's interested in that, we're happy to share that protocol. So the majority of the costs will be subsidized, but the awarded labs have a buy-in of $1,000. So if your proposal is selected to move forward, the commitment from the lab financially is the $1,000 buy-in. And so again, we sort of encourage anyone who's interested in submitting a proposal, or even if you're awarded if you're made the award, then to reach out to us and we can chat about the logistics of sample prep and all of these things to make sure that you're on track to meet the deadlines that we need, both scientifically and financially. We had this up here before, and I just wanted to sort of reiterate that the information about the technologies, all of these things are listed on the website through Yuri's core. So feel free to go to the website and check these things out if you're interested in more details. I don't know why this is doing this, but I just have some additional questions that people had been asking that I put up here. I don't know if there's other questions that people have, but we had been asked about who's going to be judging the proposals. So essentially, as part of our initiative, we're really going to reach out, obviously, to people within BU to judge the proposals. This includes winners from last year, so they're going to now serve as judges this year. Again, how long should the proposals be? I had mentioned this, 2 pages maximum. We're not looking for, massive amounts of preliminary data, just enough to rationalize the science. Again, exploratory is fine. It doesn't need to be hypothesis driven. And can it be collaborative? Of course, obviously, we like to support people who are working together in this. So if you have a co-PI kind of proposal, that's fine too. And then again, this is I think the most common question is people who are saying, I received either a GSI seed grant or I was awarded a single cell pilot last year. Am I eligible this year? So we're not saying you're not eligible, but we're going to give priority to people who haven't had the benefit of support from the GSI in the recent past. So feel free to submit, but we can't make any promises. So I'm not sure if people have other questions.
With the proposal length, the 2 pages should be the science and the figures, but references aren't part of that. You can
Sure.
Some people have asked if the references
Oh, really?
are included.
Okay, references not
So
included.
references
So
not
yeah.
included. Yeah.
So you mentioned live cells. So this technology requires
Viable.
that would provide viable cells for core, or will process them to
You
prepare
can do single
nuclei.
nuclei, but
Without fixation. or
either
anything.
live cell or nuclei. Yeah, no, people do use frozen cells and fixed cells, but we don't have any experience with that, so I
Mhm.
it would be at your own risk.
Look, so take live cells, prepare nuclei.
Right.
Yes.
Basically, viability is indeed key to success. If your cells are 90% viable, you get a very nice partition of these cells into those droplets, and you get good data. If it's less than 90%, it just becomes a very expensive bulk experiment.
And if people have questions about the $1,000 buy-in, that's required for covering the bioinformatics.
Computational piece. Yeah, I should have mentioned
Yeah,
that.
the bioinformatics for the single-cell sequencing core. There's still someone that has to take the data and
Put it through the pipeline.
put it through the pipeline.
And so the analyses are all going to be sort of standard analyses. So if the
Yeah.
expectation is that you want something beyond the standard cell ranger, that kind of thing, then that needs to be, that might be something that you would have to forage on your own with additional support. But that's not something that's part of the pilot.
But
list.
having said this, our standard analysis improved quite a lot since last year, so we'll be producing a pretty comprehensive report on this.
Right.
Yep. They were the only comments I had.
Any questions?
Are there any questions
Thanks.
online?
I actually don't know how to use the online.
I was gonna ask
chat
that. How
feature.
can they-
I
people
just know
ask
there are
questions
10
online?
people here. I haven't
Okay.
figured out how. We'll figure that out in the next version of the live stream.
Yeah,
But
right.
if you have a few minutes to go over the Loops
Yeah,
browser,
sure.
I'll just see
To step back up again.
a little bit about how the bioinformatics visualization is.
You have your adapter, right?
Rosh, I've got to run. Thank you
Good
very
to go?
much.
Okay, see you later, Rory.
Thank you. Just like 15 minutes on the browser.
Yeah, probably won't even take that long. We'll
Okay.
see.
I know, I'm just looking at that.
It's very exploratory, and we have
Is it?
examples online too that takes you like step by step. So,
Oh, okay.
yeah. So when you download Loop browser, these files are already these C-loop files or whatever you would upload to Loop Cell browser already comes preloaded.
Yeah.
So these are example data sets you can easily play around with. So we will do the ATAC tutorial.
And you have it on the. Can it run on a PC or is it only on Mac and Linux?
So Loop browser will run on PC and Mac because
Okay.
it's just a visualization tool.
Yeah.
When you do cell ranger analysis, that's where you need the Linux environment to be able to run the pipelines.
Oh, okay.
Yep.
Okay.
So here, every dot is a cell, and the closer they are, like they're more in terms of accessibility, similar to one another. So for example, this cell here is closer to these here compared to what you see here. Again, this is also t-SNE plot, but it's also 2D, so you kind of have to keep that in mind. If you looked at this in 3D, some of these things might not actually overlap in 3D space. But essentially, this is Loop Cell browser. You can look at your data in a few different ways. So categories is where we're at right now. Graph-based clustering and k-medoids. These are just two different kinds of algorithm where you do principal component analysis to reduce your dimensionality. So graph-based we usually use by default. There's a bunch of tools on here. You can save photos, you can upload file. There's also this option called split view. So it just splits all of these different clusters on different panels. So if you just wanted to select a particular cluster, it's hard to do that when it's overlapping with other clusters. You can do that as well. So one example I'll show you is, so if I go to accessibility, and these are PBMCs, so for example, I look for CBP alpha, EBP alpha. So CBP alpha is a transcription factor that's expressed in monocytes. So you can see, it's a little hard to see, but most of these up here are red, most of these down here are blue. So if you wanted to label these cells and let's say call them monocytes, you can use this lasso tool. Oh gosh, it's hard to do on that screen. Let's try it here. We'd use this lasso tool. So let's call it cell type, monocytes, save. So if you go to cell type, now we've categorized this as monocytes. Now we have multiple other clusters as well. So I'll go back to accessibility and look at CD3D. So this is for T cells. Let's delete that. So now this cluster down here is what's lighting up. So I want to categorize these as T cells. So I'll select these.
So this is pretty arbitrary by the investigator. You just
Yeah, it's just a lasso
circle
tool, so you're just like
lasso.
drawing around the thing. So cell type, T cells. So we'll save it. So we have monocytes and T cells. We can also look at accessibility peaks. So this is because this is ATAC data. So if you go down here, there's a peak. Viewer here, so you can either give it a region or a gene. So let's just look at CD3D. So you'll see monocytes, T cells. This just looks like a block. So what you can do is when you run your Cell Ranger ATAC pipeline, it generates something called I don't know if it will show on my screen or your screen. Okay. Loop. There's a fragment file that it generates. So you just have to load that fragment file, and now you get these peaks that you're probably used to seeing for ATAC data. And one of the things you can also do is, if you go on here, you can export this as bed graph. So if you wanted to look at this on IGV or UCSC Genome Browser as your single cell data peaks, you can look at the bed graph files on there as well.
Wait, sorry, how do you connect the ATAC-seq to like the gene express, kind of the RNA-seq?
So for right now,
single cells, or
so right now, you know, because we haven't released the product, we don't have a software that does that. For what other folks are doing right now is they can use Seurat. Seurat has a vignette for integrating single cell gene expression data with ATAC. There's a nice webinar that Raul did. So a lot of people are using things that are not directly coming from 10x, like a third-party tool to do the analysis. So you can see, you know, T cells have this peak here, monocytes don't. One of the other things I can show you is in terms of filtering. So if you go back here, there's this population of cells that's left. We don't really know.
What it's going to do is it's going to take each population and put a different panel for each population. Is that right?
So you're looking at different genes or regions here. You'll have each of the different populations, and it just shows you which of the populations has the peaks. So you can see the differential peak between the different populations. So we don't really know what this is, right? So we want to figure out what this is. So to do that, if we come back to categories and all of these are selected, we can then go here, and then it says globally distinguishing, hit the calculator button. So there are a few different options: motif, peak, promoter sum. The way ATAC-seq promoter sum is defined is if you have a peak that falls 1 kb upstream of a known TSS or 100 base pair downstream of the known TSS, that peak is called the promoter sum. So if you're just interested in looking at TSS accessibility, you can use that. So when you do globally distinguishing and hit the calculator, what it's doing is it's calculating differential accessibility between all these different subpopulations you have here. So this takes a little bit of time, but I think it's done. Okay, so if we go here, on this table, it gives you the different genes. We have the 3 different clusters that we were looking at. So this unknown is what we were interested in. So BLK is actually a B cell marker. So, you know, based on this, and this is just an example, you could have multiple different markers you could look at. So I'd say, oh, BLK is actually pronounced in this population, so it's probably B cells. So I will just label this as B cells. Okay, so now we have these 3 different populations that we were able to discern. And then one of the other things you can do with this tool as well is there's this option called filter. So you can, if you wanted to look just at the B cells, but you wanted to look at subpopulations within the B cell, you can create rules. For example, what I'll do here is I will say in cluster B cell, greater than 0. So TCL1A is a gene that is expressed in immature B cells but not mature B cells. So if I said look in B cell clusters where this promoter sum is greater than 0, it gives me that there's actually 29 barcodes. So if I click on this, now I can do subcategories. So instead of B cells, I can call it immature B cells. Oops. So within B cells now we have, if we take these out, you can see there's 2 different populations, right? And then I wanted to categorize everything else that was left, so if TCL1A is 0, these would be mature B cells. So I can go back here, cell type, mature B cells, save. So if you come back here now, there's, instead of just B cells, we have categories within the B cells. So you can do this with multiple markers. Yes?
If you are interested in CD4, which also is expressed in macrophages, and also in T cells, but also in NK cells. So you have 3 so far different groups.
Yeah.
How can you display the CD4 in all these cells?
So maybe go here, we'll look for CD4. So there's different populations where CD4 is expressed. So if you wanted to, so you see it in T cells, you see it in monocytes. One of the things you can do is you can go in the split view and just select what's expressed here, 2, 3, generate 3 different categories, and then you can compare them to one another. So it's going to give you a list of genes that's different within these populations, even if it's expressing CD4. So there's a lot you can do and play around, like you can add multiple different genes for doing the filtrations. What I showed is just very basic, straightforward, quick and easy example. So like I said, it's very exploratory. You can go around and play with it, and there's quite a bit you can do with it. That's pretty much what I wanted to show. Like, there's fun things you can do with it.
Cool.
Yeah. And then this, if you go on the support website for single cell ATAC, there is a tutorial that walks you through something similar to this. So, you know, if you want a refresher before you play around with something, you can do that as well. As well as what I will say is a lot of the data we've generated in-house, so maybe you're interested in mouse brain, we've done ATAC sequencing on that. These C-loop files for those data sets are also on our website, so you can easily download those and play around with those as
So we
well.
should put like a link on the GSI website for some of these
Yeah,
things.
we need to get a link because I think even on the single cell sequencing core, their outputs is this HTML based file that it doesn't link to the Loop browser,
The Loop browser. That's why
to
I think
play
it's
around
there.
with.
Yeah.
Yeah, so if you go to support.10xgenomics.com, each of the support section is divided based on an assay. And if you go to data sets, it has the raw files, output files, C-loop files, all of those are on there as well.
Okay.
All right. Great. Well, thank you very much,
Thank
Rosie.
you so much,
welcome.
Rosa.
All right, I'm going to turn off the stream.